Cool, so I'm going to present my CST, Comité de Suivi de Thèse, and it will be a 25-minute presentation, and here's a recording of the presentation itself. Cool, so the title of the PhD is Gene Network Inference via initially novel neural architectures and now single-cell RNAseq foundational models. That I do with Laura Cantini and Gabriel Perret, who are my co-PI at Institut Pasteur and ENS Ulm. Quickly about myself, I come from an école d'ingénieur in Paris. I'm French. I've done an internship at the Flatiron Institute a while back, which got me really into science and machine learning. I got into genomics at the University of Kent during a second Master of Science, and I built a company for a year with friends. I spent 3 years at the Broad Institute in the Cancer Data Science group, and I led the target discovery team at White Light Genomics for a year. And I recently, in October, joined Laura and Gabriel for a PhD, thinking it was a great project, and it was also a bit now or never for me to get this diploma. Cool, so just a bit about the topic in itself, right? Single-cell RNAseq, you might know about it, but it's really a way to get at cell by gene matrices where you get the expression of every gene you'd hope in the cell for every single cell in a tissue that you sampled from, and the expression of a gene tells you about kind of what the cell is doing at a specific time. It's the expression of RNAs. And not of proteins, and RNAs are what the cell will use, some of them at least, to make proteins. There's actually a lot of issues in these types of data sets. You have high dropout, almost like 90% of the genes that you won't be able to sequence. In some instances, a high batch effect as well across groups and tissues and samples. And on the other side, you have gene regulation, which is a very complex biochemical process, which is really how the cell does what it does, it's kind of the computation of the cell. And it's highly simplified in the field, where we simplify it as a network between genes or proteins. And this gene network, we simplify it even more by just having connections between transcription factor to genes, just kind of pairwise connections. You have simplified it as a Hill function that defines how things interact, or ODs and whatnot. And most of the time, people generate these graphs from something related to co-expression. It can be weak learners, or it can be kind of related things that will define kind of a matrix of connections between genes, and then doing a subsetting of this co-expression using, for example, only transcription factors, or additional data like ATAC-seq, or the literature data of connections between genes. And they do that over oftentimes one data set in a specific tissue or in a specific sample. Another big issue is actually that gene networks don't have a good ground truth. Most of the time, people are using ChIP-seq or knockout, deactivate a gene, and you see what's going on in the cell a few hours later, which is a long time after actually what you've done. And so many things happen in the cell, and ChIP-seq is actually very noisy, have biases, it's very scarce, and don't do this at the single cell level most of the time. So actually we're lacking a good ground truth, or a good notion of a ground truth for this type of question of gene regulatory network inference. However, you could be able to view it in another light. You could say, well, we don't know, we are not really able to see this complex graph, but we know that something that has this complex graph somehow in memory should be able to perform really well in many different tasks. Those tasks can be cell type prediction, they could be cell fates or trajectories, differentiation inference, they could be predicting knockouts or knock-ins, they could be batch effect correction, prediction even, or denoising or zero imputations. If you have a good graph, you should be able to do these tasks much better than you would if you don't know the graph of connections between genes in your cell type. Great. We can talk more about this, and if you disagree, feel free to comment. But I like to call this model large cell models, and I believe, and they've been said to hold promises at least to solve some of these issues and to solve some of these tasks. And they are pre-trained on tens of millions of cells. They look like large language models in language, and once they've been pre-trained using one simple task, can then be fine-tuned to perform a lot of different tasks, which I've given examples here below. And actually very interesting, the way they work is that they have to represent genes using and the genes expression using some vectors that you have to learn somehow or to find, and they will update these representations through the different layers of the network, and they have this mechanism of self-attention, which I will explain. So how are these large cell models work? So they use a transformer architecture, right? And they will generate actually a contextual gene by gene matrices via something called the attention mechanism. So let's dive into this, right? So you have to get somehow, most of the time they are learned, an association between each element in your input. It could be genes, could be some words, to a vector. And you will then, through two different steps, the multi-head attention and the feed-forward, update these vectors. And you have skip connections to really update those vectors and add to the initial vectors your new vector, your new update vector. And you'll do that across many layers. So the feed-forward is most of the time a multi-layer perceptron, simple neural network. And the multi-head attention is really a way to update one vector according to a linear combination of every other vectors. So let's dive a bit into this, right? So most of the time you'll do the multi-head attention, meaning you'll compute this for different subparts of your vector, an attention mechanism that you'll then concatenate once you've updated it on your vector, right? So you don't do it just once, you do it multiple times in one layer, and each of those times are called heads. I can explain this a bit further also if needed. Just a quick recap, why this is a key equation of the transformers, right? The attention mechanism. So for each of your vector here, it could be a vector 0, right? I call embedding here. You generate a Q, K, and a V, right? A new version of those vectors using an MLP most of the time. You actually compute K will be the keys of these vectors, and Q will be the query. And the query will actually be multiplied with the key of this vector and every other to get at a value that will be your multiplicative values of your linear combination of all of the V terms. And so that's how you get to this Q times K, right? Or in this equation, right? Here, how you update your embedding using Q times K for all of the I elements, here all of the input elements, all of the genes, for the query here, that you'll get a value. You do a softmax because you want to make it sparse. And if you view it across all of the input values, or here all of the genes, you'll get basically an attention mechanism from all genes to all genes. So you get a gene by gene matrix in the end. And you get that across all of the layers for all of the heads. So it can be hundreds of those actually gene by gene matrices. So that actually makes also this model very powerful for manipulating gene regulatory networks. And that's kind of some of the idea that got us into using transformers, right? And for this question, they are not like graph neural nets, but transformers, because the transformers, they don't use a graph, they generate a graph, and they generate many of those by default. And also they've been shown to train across they have these foundational models that are trained across many data sets. And so you get from an under constraint problem where you have only a few tens of thousands of cells in one data set to predict something that's very complex, the gene regulatory networks, from scratch, to something that has learned across tens of millions of cells across many contexts, to learn this now still 20,000 by 20,000 graph or matrix. That is the gene regulatory networks, or the gene networks. So this gave us the goal in the end for this project, which is to leverage large cell models for GRN inference. And the corollary of that would be how to make these LCMs more explainable using gene regulatory network inference. And so that's how I came up with scPRINT. which is this new large cell models I created for single cell gene regulatory network inference. Why did I create a new one? First, because actually the current models don't really do good GRN inference. Even though they claim and they talk a lot about this, and they perform many tasks that I explained that are related to gene network inference, the way they look at it is actually pretty scarce and not that good. GeneFormer actually might be the most interesting. They just remove one gene in the input and they look at how that changes the embedding of genes at the last layer. And the embedding of genes that change the most would be the ones that have that are being impacted by this change. However, they don't really prove it that well. They do one experiment in a cell, which is really nice actually, but it's not very quantitative. And they actually they show this one example for the GATA4 target, and it's barely significant, and it's actually not super convincing. Although the idea is nice, and I talked with Christina, who made the work, and she said herself that likes a lot of work in this domain. scGPT, which is another famous model on the topic, actually does, to me, even worse. They show it by, they actually look at the attention matrix this time, matrices. However, they only kind of look at them by giving cells that have been perturbed with the actual expression of the gene that you want to look at. So here, for example, perturbed with GATA4, and you look at the difference between the control and the perturbed cells in the attention matrices. So you take the attention matrices for the control, for the perturbed, you do some kind of rank normalization, a binormalization actually on those matrices. I don't know why not. And then you do a comparison between the one and the other. And those gene connections that have changed are like elements of your GRN somehow. But again, no quantitative analysis, no benchmarks, and it looks a bit like sortie du chapeau, as we would say in French. So not that great, but gets even worse, because actually I would still love to have used those models, and I started to do that actually with GeneFormer and scGPT, but I stumbled upon many other issues that have also been, some of them explained in two review papers. First, for me, they're just not easy to use, even very hard, not really coded well, feels like it lacks a lot of things. I don't think it's made on purpose, but it's really not meant to be reused a lot. Also, they have to be fine-tuned for all tasks, and I think that's a big issue. And in the end, it's even shown that the pre-training of those models, the reasons why they're here, basically they're being pre-trained, that's the important thing, doesn't help here in any way. So there is a good example in the paper of predicting knockouts, and you see that they do well, not as good as the state of the art, and that's the case for most tasks. They don't do as good as state of the art, but the difference between the trained and the pre-trained and the non-pre-trained is super small, and the zero-shot prediction, if you don't do fine-tuning, is really, really bad. So that's a big issue, and begs the question of, is the pre-training task any good? And when we look at it, and it did not convince me a lot. Removing 15% of the genes, the way they are predicting expression, there's a lot of things to rethink, and so that's a big issue. But even I would have tried to retrain these models myself, but there's actually no code for how these models have been pre-trained, how to get the dataset, how to load it in memory, how to do it across GPUs. There's nothing about this, so it's a very weird version of open source. I'd say it's open inference, it's not fully open source. It's open weights, but it's not open training. So that's how I came up with scPRINT. I try to solve many of these issues. I won't go into so much details about all of the updates that scPRINT brings, and then kind of a response to the things that scPRINT is really geared toward, kind of making updates for also gene regulatory network inference. So in the pre-training tasks now we get information of protein language models. I will go into that a bit more. We include the gene location information in the DNA, so that you know that genes are close together, might be regulated by the same set of transcription factor of CREs. The expression is actually parameterized by a zero-inflated negative binomial distribution, like you've seen in many other many other like single-cell RNAseq models. And in the pre-training task, there's quite a few different things, including classification as part of the pre-training, because we have good classification information for most of the cells in our datasets, actually. And many more things. One key element actually is that we do weighted random sampling, which has not been done in the other model, and I think it's a big issue. Right? You have lots of sampling bias in the data. Most of your cells come from the same data sets, the same tissues, and they are the same cell types. So you need to actually weight your sampling when you do the pre-training and you create your mini batches. So one key task is a pre-training task, right? It's how you train your model from the beginning. And we actually came up with a new version of the task called denoising. And in this task, instead of masking genes with a masking embedding, we actually subsampling the gene expression. So if the gene expression has a sampling of total counts of like 20,000, we are subsampling it to 10,000, for example, and we are asking the model to recreate expression as if it was 20,000 sampled from 20,000 counts. And so we are dropping genes, we're having zeros, we are decreasing expression, and asking the model to recover this, which is actually related to even the data that the model sees all the time, right? The data is subsampled in itself, it has dropouts. So I think that's a better pre-training task, and it's somehow actually, funnily enough, related to DDPEM, so denoising, diffusion, score matching models, although it's just related to. It's not the same thing. We're doing it in one step. We don't sample from complete noise, and we are not doing really a generative approach. It's still an encoder kind of like model. Cool. So maybe we can focus a bit more on this gene embeddings things, right? So the goal is you have to encode genes, like say this is MIG, this is MIB, this is something else. And how do you do that in those models? This is not like in VAEs or simple MLPs. You don't give a value, you give a vector to the model to tell the model this is this gene. And in this vector, you tell the model about which gene it is and how much it is expressed, and whatever you want has to be contained in this vector from the input. So here in our case, we're actually using a protein language model to encode the string of the protein associated with this gene into a vector. So a protein language model will encode not only the string, but information about the protein, like its binding affinities, stuff like that. It's really key and interesting for the gene regulatory network point of view. So let's dive into this, right? So these models, we take ESM2, and this is extracted from their paper, and it's how the model is trained. So again, a transformer trained with masking. You have the sequences, and it has to recover it, and that's a pre-training task. But there's something much more interesting, right? So it talks about transformers. The title of the paper is Transformer Protein Language Models are Unsupervised Structure Learners. And so what they say actually is that once a model has been pre-trained, you can extract the attention matrices, perform a few like processing steps, but not really important here, and then use linear regression with a strong L1 regularization to select a set of attention matrices that contains or should contain the information about the 3D structure of those proteins. So although the model has never heard about 3D structures of proteins, it's just recreating sequences of proteins. They found that actually in the attention matrices you could recover pretty well the 3D structure of the proteins. You had this information embedded in the model. And the regression is not really used in the end, right? They just use it to select the right heads, and once they have it, they just take the mean of those heads. And that's really interesting. And the last point is actually, in addition to showing how to extract kind of this structure from the attention matrices, they also show how scaling, you had a scaling law of those models, and as the bigger it gets, the more information about the 3D structures you get in those attention matrices. So that shows that brings me to the current results we have now with Sprint. So we've been able to pre-train Sprint and get some early results and comparisons. So here's not the same slide as before, it's just the same plot. I really just did from ESL2 and just updated a few things to say that in our case, well, we don't train with masking, we train with denoising, but we have also an expression database, we have this pre-training task, and then we can also extract attention matrices across all of the heads and the layers of the model. And instead, here we might do some kind of a classification, like with a strong L1 regularization. And here we would use maybe a ground truth gene regulatory network to try to classify and learn which heads contain information about this GRN. So we can use that, we can take the max of those heads or the mean, and that's a way to extract this information. And actually, in our case, because cells are very noisy, we want to take multiple cells, and the way we compute these attention matrices over maybe 1,000 cells of the same cell type, so 1,000 neighboring cells, is by computing the mean over cells of Qs and the mean over cells of Ks, and computing the attention matrix over those aggregated Qs and Ks. So that's just a quick updates. And once we have that, we have actually a genome-wide network, a gene network extracted that is cell type specific from 1,000 cells that is 20,000 by 20,000. So that's really great. So let's look in the results. Once we have this GRN, like we have in our case 2 ground truth that we've selected. One is OmniPath, which is non-cell type specific, just from the literature, but it's quite big, the biggest available. And we have a dataset from Sushmita Roy that is aggregating chip, seq, and knockout data from the same transcription factors, over a dozen, from embryonic stem cells of the mouse and human. And actually, we can see that we perform better than what we would call the state-of-the-art here, which is not that great in the end. It's Gene3, which is quite simple. It's really XG boost that you're using to predict each gene's expression according to every other, and you do feature selection, and those genes and those features and their importance is really how you make those connections from each gene to the others. That's pretty much it. And then that's actually what's performed best most of the time, according to benchmarks for single-cell RNAseq, gene regulatory network inference. And what we see, and I really should change this curve, it's not that great. There's not really such a thing as random recall and precision recall. So the random curve, like the red curve here, please think of it as just a random line that hits at this level here at around 0.0045. And yes, the precision is really low, the recall is even worse because it's a log scale. And Gene3 performs a bit better than random, although not that much more, at predicting OmniPath data from, here it's a random set of cells expression. I gave the same random set of cells expression from one dataset only to SPRINT, and we get much better results here than Gene3 at predicting OmniPath data. However, you know, there's 2 versions of Gene3, Gene3 regular and Gene3 where you only look at transcription factors. In this case, you help it quite a lot. And if I were to show you Gene3 on transcription factor here, it would be 10 times better at than SPRINT. It would go at around 0.06 or 0.05. But you have this bias, and I can't really yet compute well the random, like what would be considered random in this case. There's another dataset where the Sushmita Roy, and here actually SPRINT performs really, really well, much better than even Gene3 TF, much better than random, and these values are actually EPR, which is another metric that is actually quite used in the field as well. And I should also show EPR for OmniPath, I just don't have it here. Cool, so there's a lot of buts in these things actually, and I will talk about it a bit afterward. I have a few questions for you also, and you'll have a few for me, but I can also show that those models, the model SPRINT is really meant for GRN, but it does many other things. Like it does classification, batch correction, denoising. Actually, I won't go into the result that much, but yet to be great. Still a work in progress. It's with old version of the models also that were not that great, but the results are also subpar to what exists, what would be considered state of the art. And so there's more work to do, but we also already, what I want to show here is we already have the model and we already have the capabilities to benchmark it. The paper is not a benchmark paper, so we won't benchmark it against everything, but that's really kind of the four main topics that we'll be looking at. And if you have more question, I can present that a bit further as well. A last point is we're actually trying to show scaling laws as well. So we are training different sizes of models. The biggest one still have some issues and will need likely more GPUs, but will be state of the art in terms of size. We are still already seeing scaling laws in terms of the losses, denoising loss, and classification, but there is also more work to really make it comprehensive in this regard. But we do, I think we'll be able to do on par with the other models in terms of size, and that's already quite great. And there's an open question: is why do we do that much better than them in terms of compute? Like we don't need as much compute to train that size of models, and I have a few ideas for why. Could be related to the weighted random sampling, to the zero inflation, negative binomial that we are doing, and all of these inductive biases that we give the models. Cool, so there's already quite some work that has been done, but there's still some more things and open questions. And the big issue is that actually the ground truth that we have for GRNs are not that great, and that's really posing a problem on our end, I guess. So I've shown at the beginning that we really want to make the point that we don't have good ground truth, we might never have. We'll use what we have and show some results, however biased they are, and present some of the reasons why we don't see that great results for some of these ground truths. And we hope to present the idea that actually orthogonal tasks like classification, like denoising, like perturbation prediction would be good tasks that would replace these missing ground truths. But I'm open to ideas for ground truth here, and to your questions and remarks on this. Other issues is that it's a lot of work, especially if we go and dive a bit further in those other topics, denoising, batch correction, cell type inference. Cell type inference is quite easy actually, but the other two, and it's actually hard to compare to a CGPT engine for the other models, because they are so hard to run, because they are missing code, and because sometimes I feel like I need to improve them by giving them the ability to do these tasks, and we've actually seen that they perform near random in many of those or really badly in many of those contexts if they haven't been fine-tuned. And in my case, I don't want to fine-tune the model I have. Cool, so that's it. Basically, I'll share the slides with you if you want to read everything, and then there's a few more slides afterward, after the questions, to know more about these tools. I've already released 3 packages. The data loader for the model that shows you how you get those big data sets, how you load them in memory, how you do weighted random sampling, and it's been released to the public. BenchGRN to basically benchmark generatory network inference tools from single-cell RNAseq, and GRN data, which is a version of AND-data to work with generatory networks and single-cell data. So a few more work before being able to publish, 4 key points, I guess, but I'll leave you with also this image that shows that I generated a few days ago, that shows the UMAP of the embedding of cells from scPRINT, together with a classification of the cells from PRINT. Actually, it's just the 42 most predicted cell types. And the arrows are not pointing where they should be exactly. That's just because of some bias. It's the 42 most predicted cell types, and there is other things that impact the embeddings, like disease type and other classes. But you still have the idea with the colors, right? You have the pancreatic D cells, it's in green, you have this big green blob. The blues are the double positive alpha beta thymocytes, the hematopoietic stem cells, the lymphoid progenitors and whatnot. And all of the here you have all of the brain cell types. So you still get quite meaningful representation, and that's quite cool. Well, thanks for your time, and happy to read your questions if you have any. Bye.